It's the first time I'm speaking in this time of forum, and I hope I do a good job of showing you the data and explaining without going into jargon and so on. If I don't, please raise your hand and tell me that I have I need to explain myself. I don't normally think about these issues on a regular basis, although in some ways I do. And so this has been a very interesting experience to me to sort of review my life as a scientist under this particular lens. So what I'm going to talk about today is incidents where irreproducibility actually proved to be new data, or hiding data, or some other aspect in the scientific or in the experimental system that we were not really considering when we set up the experiment. And I don't think it's a flaw science. I think it's part of science, and it's something that we need to recognize and something that we need to pay attention to when we perform experiments. So I work on protein folding and protein folding diseases, which are also associated with aging. And I do that, and I put the question in the context of multicellular organisms. And there are all kinds of models, system to look at multicellular organism. And there are many advantages of using such models when you do research. The only problem, or let's say one of the main problems of working with multicellular organism is that the complexity of the question and problems that you're facing is sometimes very high, and a lot of time you treat the experimental system as a black box, and the things that comes out of it are not always very clear. So I work with C. elegans. It's one of the simplest model organism, and it's great in a sense that it has a lot of advantages that we can use, specifically if you consider reproducibility. So first, they are highly conservative, reproducible animals, meaning that you can trace each and every cell in the adult animals, and you can know exactly the lineage of where it came from. So we can track each cell division, and we know that this particular neuron came from this cell, from this cell, from this cell, all the way to the first division of cells in the egg. Okay? So this is a great model for developmental biologists because you can predict what a cell will be in the next division. And so you can learn a lot about developmental biology. It is also a very good model for aging. They live short time, and the best advantage is that they are hermaphrodite, meaning that you put one worm, you get 200 worms 3 or 4 days later, depending on the temperature, and those are supposed to be genetically identical, okay? I insist on supposed to be, but they are, for the most part, the most genetically identical model, meaning that you can take this population and treat it as a different, or as an experiment that repeats on the same population. So you do biological repeats, but it's supposedly the same animal, reducing a lot of the variability that's associated with other strains where you have to take a male and a female in order to get a population, and you introduce genetic variability just by that. And one more advantage, if we are talking about it, it's transparent. And so that means that we can look inside and look at living animals and see exactly, or follow what's going on inside there. So when we work in this model system, and I think it's probably, or I assume it's a general concept when people working with a model system, is that you can work in different hierarchies or levels of complexity. And each level can give you different things in terms of what you're trying to do from there or get from the experience. So the first level is the organismal level, mainly and majorly by behavioral assay, where we take the worm or whatever animals it is, and we check what is happening to the worms. And Cindy Brenner, that introduced the C. elegans, actually brought it into the lab because it is an extremely simple model where you can take a gene and translate into function or a phenotype, and that gene-phenotype connection can allow you to solve the problem of gene phenotype. In fact, if you look at the gene in C. elegans, most of them are named by the phenotype that they're showing: uncoordination, dumpy, long, age, so on, and you will see some examples. And so behavior is a very strong, it's a very strong assay, or behavioral assays are a very strong experimental approach, because they allow us to do a sweep and very quick way of looking at the system and finding interesting phenotype or interesting questions we want to follow. But it's a black box. We don't know, even if we assume, what exactly is giving us the phenotype and underlying mechanism that might impact that phenotype. And a lot of variability and irreproducibility, as I will show you, come from that. The second level is the cellular level, where we look at protein or cell component using various techniques of staining or microscopy, where we actually trying to understand which tissue is showing which phenotype, and to understand the organization of the protein in space, or where they are doing their work. And the final one is that we can always treat the C. elegans, or let me say sadly, as tubes that contains macromolecules which we can purify and then study. So we can study the proteins, we can study the RNA, fatty acid component, and we can ask about their level, their composition, and so on. So this is an example. I will come back to this example because it's one of the examples I want to show. But this is an example of how you can sort of see this principle in action. So we looked, or we knew that protein folding environment change when the animal is aging. We all know that system deteriorate with age. And so I wanted to do an experiment to ask when the change occur. And so I used a simple assay of surviving stress. Okay, a stress response is an acute response, and it requires a lot of quality control. And surviving the stress is a good indication of the quality control networks actually working properly. Okay, so it's a very simple experiment where every day you take similar animals, not the same animals, expose them to similar stress, and ask how many survive. And you can see that the first day of adulthood, all animals survive. But to our surprise, already on the second day of adulthood, this pattern changed, and we saw what now called in the literature collapse of proteostasis. Okay, dramatic change in the ability to activate the stress response. As you can see in this work, we also find mutants that don't do that. They are very interesting because they are associated with the reproduction system, but a particular aspect of the reproduction system, I will not go into that. So this is on the organismal level. Then going into the cellular level, we can use a reporter. This is a fluorescent protein that is under the regulation, the genetic regulation of the heat shock response. And we ask whether or not it is changing the expression. And we can see that on the first day of adulthood, you get a heat shock response. This protein is produced. But on the third day, it is not. And our mutant corrects this change. Okay, the last is we can take the worms, we can purify RNA or protein, but this is RNA, and ask whether or not RNA level change. And you can see that the wild type RNA levels drop dramatically, but our mutant stays or shows similar levels. So this is basically allowing you in some ways to do reproducible experiment, but using very different techniques and different levels of interrogation in order to actually say that this is what you're looking at and not just something random. But even when doing this, things can be affected. And what I want to do today is show you several examples, these experiments, but also a few others and some from the literature, where sometimes it is reproducibility, sometimes it's variability. And I think if I want you to take some message from this work is that complex systems have a lot of layers that you have to pay attention to. And in those layers, there could be hiding either new truth or really flaws, big flaws and failures. So you have to pay attention. I'm going to go and give example to environmental factors that can contribute to this. And I will talk on genetic factors. And I'll give more example here because some of the genetic factors are hidden because we don't really see. The animals doesn't go with like a note saying, this is my genetic profile. We're sort of guessing it. And a lot of failures come here. Okay, so this is almost the first experiment was done in my lab here in Ben-Gurion University. I was a new researcher, and we did this experiment, and we saw this collapse, and we were extremely excited. I mean, you know, it's like the first day of adulthood, it's changing within hours, you get this collapse. It's not like a mild effect, it's like 80% change, very robust, very exciting, okay? And we already had the mutant that rescues this, so this concept was highly exciting. I actually asked my student to sit down and appreciate the moment where her theory was actually correct, because it doesn't really happen in science very often, where you have an hypothesis and you're proven right. My student got really excited, and because as we were told here, every experiments have to be repeated, and have to be repeated rigorously, I sent her to redo the experiments and come back with the result. And as it is, she comes back a few days later and tells me that it's not reproducible, meaning she sees the phenotype, but then she repeated the experiments and the phenotype is completely gone, the worms behave completely like the first day of adulthood, and there is no collapse of proteostasis. I'm a firm believer that if you see something and it's substantial, it's usually there. I say usually, and so one of the things that I've asked her to do is let's go and look at the raw data and see if we can find something that can explain this behavior. And this is what we noticed, and you can see it summarized here. When she had experiments where she was using small amount of worms, she saw the phenotype. And when she did the experiments with a lot of worms, the phenotype was gone. Okay, so now you can look at this data and you can say, okay, something is really, really awful here. Obviously, it's not reproducible, something is weird. Or you can say maybe there is something here that we are not considering. And looking at the data, we could think about two things that can play a role. One is the crowdness itself. It's known in C. elegans that when they're too crowded, there is a hormone that can tell other worms, it's too crowded here, let's go somewhere else, okay? And the other thing is that plates that had a lot of worms also had very little food. They still have food, but they had much less food. Studying crowdness in this particular point was not very simple. You had to produce the hormone. It wasn't a simple procedure. So we decided to test the first hypothesis and see, the second hypothesis of whether or not the amount of food is impacting our research. And yes, it did. So if we took the worms and removed them completely from food, so now there is no food, and we checked their survival over time, we could see that there was an increase in survival, and it sort of saturated around 4-6 hours, where you got 80% survival, exactly like the wild type, okay, under this experimental condition. Meaning that the lack of food, or change in food amount at least, changed or reshaped this phenotype. So, of course, now we repeat the experiments, carefully controlling the food, and everything is fine, and we published it. And we are also very careful to report that the number of worms have to be carefully monitored, and the amount of food needs to be carefully monitored, because the amount of food impacts the experiments. I hope people read methods. I do, but this is where it is. But we also took it a step further. We actually went into this because we find it an interesting biology, and then we took a mutant that has a defective pharynx, meaning that when it eats, it's not managing to grind the food very well. And so it can sit in a pile of bacteria, be very happy with a lot of food, but actually get very little food inside. And so in this case, we give them the same amount of food, but their ability to introduce the food inside change, and we can get the same thing again. So this is rescuing, and lack of food is rescuing. And we have went on to publish that dietary restriction and dietary manipulation can actually also change its phenotype, and it's changed it in a different way than the reproduction signals. So they are not overlapping, they are doing different things. And if you put both of them together, you get even bigger change in some aspect and less in other, and it's reshaping the environment, which basically means that you can take this quality control system and change it in many different ways. And it's not just one or two, and it's very important to our interpretation, and especially if you want to go in the end to medical sciences and change and affect protein misfolding diseases. I told you, we always do it properly, or we try to do it properly. So we did the survival, we did the reporter, and we did the gene activation. And in all cases, we can see that the lack of food either by mutation or by removing the food, rescue this ability to activate the stress response. Okay, so this is an example of an environmental factors that you think you control, but you realize in retrospect that you weren't controlling. And sometimes you don't really think that something as simple as having 10 worms versus 20 worms will have such a big impact on your system. And I can assume that some people didn't notice that. In fact, I think one of the most interesting aspects in the research study is that if the parents of the parents were starved in your incubator, it could actually impact the result of the experiments that you're getting today. This is wonderful work by Oded Rechavi from Tel Aviv University. Meaning that not only do you have to control what you're giving those worms, you have to also control the ancestors. And one of the things that I really insist in my lab is that you take care, and each experiment you have to go back 2 or 3 generations to make sure that the worms lived in a perfect condition, because stress can alter, and memory of stress can alter the response. And I think it's very important to pay attention to that. So the second aspect that I want to talk about is genetic factors that contribute to a specific phenotype. And I'm going to talk about genetic background, and this is really a problematic one. Genetic interaction, which is sort of a small situation of a genetic background, and then genetic drift. I hope I can get to all of them. Please tell me if I'm running late. Okay, so this is the same experiment, but this is sort of fast forward a few years later. We were looking at a modifier, genetic modifier, called LIPL4. And that modifier was changing the way the response was occurring, and we order a strain from a different lab, and they sent us a strain where the modifier was expressed, but not in all organisms. So it will be inherited randomly. Some animals will have it, some animals will not, okay? So you assume that the best control for an experiment is animals from the same genetic background that don't have it versus do have it, and to see what happens. And we did the experiment, and indeed we got the collapse of proteostasis as we would expect in this particular strain. But then when we did the control of day 1, where everything's supposed to be okay, my student comes to me and says, there is a problem. They are now collapsing on the first day of adulthood. What should we do? So I said, first, look, we have used 10, 20 different strains that all collapse on the second day of adulthood. Something must be wrong here. And so we started by doing a very simple inheritance experiment. We took the wild type, sort of, put them with our wild type, separated the progeny, and looked at the collapse at the first day. And what we found is that 1/4 of the progeny were actually collapsing on the first day, suggesting that this is a Mendelian inheritance of a recessive gene. So the gene is there, and some of the population has, or this population have a double mutation in it that is in the background. We sent it to sequencing. We actually found the gene. And when we did the experiment with the genes that we got from the CGC that sends us or deposits the strains in C. elegans, with the mutation, we got the same phenotype. So this is a wild-type strain that was growing in the lab that had some kind of a mutation occur, probably not relevant to a lot of other things. They didn't even know it's in the background. But in our case, it changed the way the result looked like. And as I said, we always confirm things, so we also did it using a different methods, RNAi, to knock down the genes not by mutation, and we looked at the activation of a reporter to see exactly the system. So genetic background, as I said, could be a huge problem, because we usually use sequencing what will be called under the lamp. So we know there is a mutation, we sequence the mutation. We don't sequence the rest of the genome, we don't know exactly what's there. We assume that it's similar, and we assume that there are not big changes, and this assumption needs to be tested from time to time. This is why I usually backcross things into my N2 background and the wild-type background, sorry, and I use a lot of time more than one wild-type background to check if I can, and also replace the worm from time to time. All are good practices to maintain or avoid genetic mistakes. The next example I want to give is actually from the literature, and I think it's you probably don't know the example specific, but you for sure heard the repercussion of some of this, in the sense that all of you probably heard one time or another that if you want to live long, you need to drink red wine. And you need to drink red wine because they're resveratrol, and this is very important molecules that can extend your lifespan. And the reason it's such a very important molecule is it's because it's affecting the activity of an enzyme called SIRT2, which is supposedly extending lifespan. Why do I say supposedly? Because there's a lot of literature showing that it extends lifespan, and then there's literature that says that it does not. And while it's obviously affecting a lot of aspects of aging, you have to be very careful. And I think one of the most important things about being careful is about this particular area is how quickly it transitions into not medical, but consuming products and selling the general audience things that can extend their lifespan. In the sense, assuming that nobody will ever sue you because nobody would live that long. By the time you show that you died, you can't really sue, right? So it's sort of like the perfect, it's the perfect scheme area, and this is why I'm very, very careful when it comes to looking at longevity modifiers and consumer beware, let's say. Okay, so this is an example from a study in C. elegans. I am not showing you all the data. There is also yeast, there is also flies, and data controversially in most of those animals, let's say it like this. So this data, what it shows, it's a lifespan curve, meaning we are asking how many of the population is still alive. You start with 100%, you follow how many died, and you basically ask when is the last animal dying, okay? And so the typical N2, if you grow it at around 20 degrees, it's approximately 3 weeks for median lifespan. And then what they had is they had an extra chromosomal array, meaning expressing something but not everyone expressing it, of this gene SIR2. And when they did it, you could see that the arrays all extend lifespan. They were different extents, but they were all doing that. And then the next thing they did is integrated those trains into the genome, so now it's stably inherited to the other generation. And you can see that there is one strain here that really substantially extends lifespan. As I said, there's data from other sources and so on, and this has been in the literature for a while, and a lot of people trying to reproducing, and so on, this information. But then came this paper 10 years later, and I actually have to say that I really appreciated the fact that this was published in Nature too, and this was a very careful work by a lot of different labs, showing the absence of effect of SIR2 on expression of lifespan in two systems, C. elegans and Drosophila. So normally, negative data would not be published, definitely not in Nature, but in this case it was. And so what you can see here is that they have shown that the SIR2 extend the lifespan, okay? But then they cross it out, so meaning they took a wild type, as I suggested or explained before, cross it several times, and follow the gene, not the phenotype, okay? And when they did that, the SIR2 that was backcrossed, so it's genetically a mutation in SIR2, but it's no longer extending lifespan, okay? It's back here. They then found in the background a different mutation. It's a mutation affecting neuronal function, and this particular mutation is known to affect lifespan, and they were able to get that mutation separate, and they were able to show that the lifespan goes with that mutation. So basically there was a background mutation here too, like we showed before. And the background mutation was the one that was giving the phenotype. And if you did your experiments by following up the phenotype, okay, anytime you crossed two strains, you made sure that the phenotype remained, then you would say that it's extending lifespan. But in a sense, it wasn't, because it was the other mutation, or a hidden mutation in the background that was giving you this phenotype. This, of course, have repercussion in many different ways. This was answered, I am not going to show you, by the author of the original paper, claiming that they redid it and it's still showing 10% extension of lifespan. I would leave it to you to argue. Another example is this. People in worms work with worms. I said that one of the advantages you have one worms and you have 200 worms is amazing, it's really cool. It means that you don't have to cultivate the animals all the time, but one really big disadvantage of this is that when you have one worm on a plate and you want to look at that worm, 3 days later there are 200 worms there. And so you can tell the difference in the beginning because one is big and the others are small, but then of course the other grows and then it becomes really impossible to know which was the experiment worm and which are the progeny. And this is really important when you do a lifespan experiment because now you want to look at 20-something days and you want to look at a lot of different conditions and a lot of worms, and you cannot, or it's very hard and rigorous to do it when you have to separate the mother from the progeny all the time. And so people find shortcuts, and one of the shortcuts is a drug called FUDR. And when you use FUDR, you basically block the production of progeny. Very popular in lifespan experiment, and you can see that its application was justified in showing that when you take the wild type and you do all this manipulation and use different concentration, you do not see change in lifespan. They live the same. But then someone did experiment with a different mutation. This one is affecting the mitochondria function. And if you do the experiments under normal condition, and you move the worms from the progeny, and you are very careful, then you see that this mutation shortened the lifespan substantially. Okay? But if you then go, so compare this one that lives like around 12 days to this one that lives like around 20 median lifespan. But then if you put now FUDR, this strain now lives twice the amount of wild type does, and you would assume that this is a gene that actually contributes to extended lifespan, which is completely reverse from what this gene actually is doing. So this again, sort of precaution to think about genetic background, genetic interaction, and things that are occurring when you're experimenting that sometimes you're not considering. And even if it's a common practice, sometimes you need to pay attention to the properties. Okay, I'm going to give two really quick other example of genetic interaction, genetic drifts. Those are more example to think about. Do I have to finish?
5 minutes.
Okay, excellent. So I will do it in like 2 minutes. Just to say that those things happens, I'm actually not giving something that didn't work. So this is a genetic background. Geneticist knows very much about genetic interaction. I showed you some genetic interaction when we didn't know that there was a mutation in the background. What I want to add here to this genetic interaction is the fact that genetic interaction is not constant all the time. It could actually change with the organism age, tissue, behaviors, and those are things that you want to look at. So for example, if we looked at the effect of this mutant on this mutant here, we would not necessarily say that it's experimentally different. They are behaving quite similarly. But if you look at the first day of adulthood, the difference is quite striking. Okay? And you can see the difference also in the molecular level. So the fact that you tested at one time or another is not always a control, and I know there is an end to controls that you can do, but those are things that you need to sort of consider when you're looking at a phenotype, the fact that there could be interaction that are specific to a time and place. The other things I want to give is one of the our last publication, and really exciting in the sense that figuring out was really interesting, and I think it's an interesting phenomena we don't usually consider when you follow genetic changes. So in this particular experiment, we crossed two strains, and we got the mutant and we got the wild type, and this particular strain also have defective mitochondria in the background. Okay, and we did the experiments, and we could see that we get the phenotype, and it's quite striking. And then we looked at the progeny a few generations later, and the phenotype completely disappeared. Okay, so this was really weird. So we started doing very carefully monitoring each and every step, and this is what we found. The first generation, you have a strong phenotype, and this phenotype disappears. On the second generation, third generation, fourth generation, you can also see it here, and developmental phenotype. Worms barely make it to adulthood, but then the next generation seems to be fine. So what happened? Now when you look at this, what you realize that for whatever underlying mechanism was actually clearing out the defective mitochondria. There was a selection going on here. And basically what we had is that the animals that we were testing in generation 3 and 4 were very different from the generation 1 and 2, because generation 1 and 2 still had defective mitochondria in the background. Generation 3 and 4 no longer had it. Okay? And so, again, one of the things that I want to say is that you have to, in experimental system, pay very careful attention to your system and how it's behaving, and really to get to know it. Okay, so a few concluding things. First, use robust phenotype, and explore different experimental conditions. Don't just use one and be open to changes. Check and check again. To err is human. Okay, we make mistakes. The biggest problem is sort of continuing with the mistake or not fixing it. So you have to find them, and you have to do the best you can in terms of removing the mistake or owning your mistake. Use different methods, different tools, different genetic background to explore a system. Don't use one way. The more different tools you use, the more likely you are to find a mistake, if it is or a mistake or a problem in the system that you're not expecting. Look at the raw data, and then look again, because the raw data can tell you a lot of things that the analyzed data cannot. Carefully report on experimental procedure. And this I got from my PhD supervisor, and it's the way I believe in doing science, is that the data is the sovereign. What the data says is what we are sort of taking as interpretation and how we follow the result. We might have hypothesis and moment of glory when we are right, but for the most part, the data is right. So we listen to the data. And that's it. Thank you for listening. Sorry for...
Thank you very much for the insights from within your lab and molecular cell biology. Questions?
Okay. I was either very clear or...
You were very clear.
Okay, thank you. First of all, I really like your lecture, and also this subject is very popular in recent years, the thing of stopping aging. So I wanted to ask for your opinion. How do you, when you talk to the general public, so how much of, like, you cannot report all the caveats in your work, right? When you talk to or the
yeah, so you know, this is the funniest thing is that...
listening. So where do you draw the line, right? Because I know there is at least one person, I'll talk to you later, that wrote a book about it, that he really sells it, right? Really
There's a lot of people that sell it. There are a lot of people who make money out of aging.
So what
Well, I have to say, this reminds me of a very interesting experience where I was invited to give a talk in the elderly house. And I started to talk about this, and someone stopped me and said, you know, if it's not relevant to me, I don't really care. It's like, tell me how this extends my life. So I usually say, when people ask, I say, look, I can extend worm's life. If you're not a worm, I'm not going to give you any medical advice. Some of the principles we find in worms can be found, of course, in other model organisms. Otherwise, we would not use them. But we are obviously not a worm, and things are becoming more complex. So, for example, dietary restriction is one of the... I know there are a lot of people are running with this diet in intermittent fasting and so on. There's a lot of literature showing that this is correct and it's impacting different animals. But then when you look at the data, the more complex the organism, the less clear the data becomes. So it's not that it's not necessarily correct, it's just that the complexity of the system and metabolism regulation becomes harder to actually tell what is exactly the impact. So what I try to say is that I'm talking about principle, and I want to explain biological principle, and I hope that one day some of the things that I find will, you know, be related to changing in medical. But right now, what my main focus is to understand the system and how it works, how it thinks, what exactly it's doing.
Okay, thanks. We have another question from Ute.
Yeah, briefly. I understood that you consider the experiments which showed that CIR1 does not extend life are more reliable than those that show the opposite, right?
Yeah.
I find that very sad, but it has to be accepted. And second, you said data is the sovereign, but I showed before the cases where data were misleading.
True.
So you have to go also into interpretation, and data
Yes,
alone cannot decide on anything.
That is true, but this is where I said that sometimes we fail. And this is why I, in the end, think that we don't report on the proof, we report on the hypothesis and the supporting argument that we have for that hypothesis. And we always assume that there might come an argument that will make us look at the data in a different way. Yeah, sometimes you think that your data is correct, you can assume that using SEER2 was originally assumed to be the correct way, okay? But then when you find a flaw in the experimental system, I think that you have to appreciate that this is a problem and then accept it. And yes, sometimes the data is not speaking clearly. I would say it like this. It's easier when you work in a less complex system, but it's still there. It's a problem that you have to appreciate. Yeah.
Thank you very much, Anat.
Thank you.
So now we have